Genes make us who we are. But what are genes? Genes are present in the cells of living organisms. The human body has between 20,000 and 25,000 genes. A gene is a small part of a molecule called deoxyribonucleic acid, or DNA. DNA is a two-stranded molecule. It's made of a chemical backbone with 4 base pairs: guanine, G, thymine, T, cytosine, C, and adenine, A. These spell out the language of what's called the genetic code. It's a language with just 4 letters: A, T, C, and G. Now that we know more about this code, we can understand more about how our cells work. It's the number and order of these base pairs that determines who we are. Each gene is a sequence of base pairs. The order of the pairs gives the instructions for how a cell will behave. For example, your genes cause a cell to be a brain cell or a heart cell. And your genes determine if your hair will be straight or curly. The long molecules of DNA are organized into chromosomes. Different species have different numbers of chromosomes. Humans usually have 46 chromosomes, 2 sets of 23 or 23 pairs. We inherit 1 set from our mother and 1 set from our father. A complete set of chromosomes is called a genome. But just 60 years ago, scientists couldn't understand what DNA looked like or how it worked. In the 1950s, this area was being researched by a lot of scientists. In 1952, a scientist called Rosalind Franklin was working in London. She took an X-ray image of DNA. Her photograph was very important because scientists could finally start to see what DNA looked like. Meanwhile, at the University of Cambridge, a team of researchers were working on the same thing. 2 scientists called Francis Crick and James Watson saw the X-ray, and it helped them to understand how DNA works. They made a very famous model which showed the chemical structure of the molecule. This became known as the double helix. This discovery was very important because it showed how genetic information could be passed on from parent to child. Crick, Watson, and Maurice Wilkins, a scientist who had worked with Rosalind Franklin, were awarded the Nobel Prize for their discoveries about the molecular structure of DNA in 1962. Sadly, Franklin had died of cancer in 1958, so she couldn't receive the award. Today, we not only know what DNA looks like, but more and more is understood about what our genes do and how they can be changed. In the 1990s, the Human Genome Project began. Scientists wanted to identify the human genome, and they began work to determine the sequence of base pairs which make up DNA. The human genome comprises 3 billion base pairs, so the work has taken several years. When a gene changes, or is missing, it can make people suffer from diseases and disabilities such as haemophilia or cystic fibrosis. Today, the genetic information that became available to us through the research of the early pioneers like Crick and Watson, and the many scientists involved in the Human Genome Project, is exploited every day in medicine and criminal investigation. And with improvements in technology, what we are able to do with this information is becoming ever more complex. But from DNA fingerprinting to solve crime, to gene therapy to cure or prevent diseases, there are still many ethical questions about how we use genetic information. And it's hard to imagine a time when this work will be done without controversy.